I think, we're just at a very early stage and to a large extent, very crude stage of understanding how to change these processes through drug therapies or other kinds of interventions.
Well, Anantha, thank you so much for taking the time to have this conversation. For the audience at home, Anantha is the Senior Vice Chancellor for Health Sciences and the Dean of the School of Medicine at UPMC Pittsburgh. So I really appreciate you taking the time to explore this incredible topic.
Yeah, thank you
And
for having me.
of course, um, and for the audience at home, maybe just give me like a thumbnail sketch of your career and how you ended up doing this work that you do today.
Yeah, sure. Obviously, when I went to medical school, it was an inspiration that drove me to medicine. And that was really seeing a very close family member have a serious psychiatric disease, schizophrenia, that really destroyed that person's life. And that was always a very sort of both an impactful moment for me as a young man growing up and going to college at that time. And that drove me to go into medicine. And I've always wanted to then understand how the human brain works and how psychiatric disorders, um, are um, sort of essentially destroying certain normal brain functions and how to treat them and how to understand them. So that drove me to then becoming a psychiatrist as well as a neuroscientist, focusing more on, you know, really discovery science, trying to find new treatments.
Now, I think I've heard you say or seen you say that psychiatry and neurology are converging, uh, and I'd love to hear you kind of expand upon that thesis.
Yeah, really. I mean, I think, when the early days when these were um, initially understood as brain disorder um, driven subspecialties or medical specialties. It started out as a single area of, you know, neurology slash psychiatry. So Charcot, for example, was the first great neurologist, but he was also the first great psychiatrist, and Freud was his trainee. So you can see in the early days, people thought of brain as a single organ that produces various symptoms, all the way from seizures and strokes to mental illness. But I think in the early part of the 20th century, people started to separate out psychiatric disorders as not having a physical source in the brain because we didn't have the level of microscopes and brain imaging and uh, various types of technology to actually see the structural changes that are happening in psychiatric disorders. Whereas they could simply see stroke and Alzheimer's and various other things actually, with a simple microscope that they were destroying the brain. So I think that's when it started to separate out because like neurology deals with visible and recognizable brain damage, whereas psychiatry deals with, magical things that happen in the brain without anything visible. Uh, but now with modern technology and imaging and high-end structural neuroscience, we know that the level of damage is at a very different level, at the cellular level, at the gene level, at the molecular level. So I think the more we understand the brain, the less we will try to separate neurology from psychiatry.
And maybe paint me a picture of how our understanding of the brain is now converging into psychiatry. Some of the things that we now understand are influenced.
Yeah, I mean, I think brain is, as many of you know, is really a very complex organ that has essentially over a billion little microprocessors within it, which are called neurons, and they continuously are creating like complex circuits, complex information processing. And what we are learning is that those circuits and those networks of information processing have to have very, you know, precise connections. And those precise connections are what makes our brain function, what makes us think, what makes us feel, what makes us move our fingers. Um, but as we see problems with that, we're now understanding that emotions and physical expressions are all driven by the same mechanism, except that some are easier to see and others are not.
What are some of the kind of recent developments that you're most excited about in this regard?
Yeah, I think there's a lot of exciting things happening in neuroscience and understanding of neurological and psychiatric disorders. I think the first one is the concept that these units or neurons are actually very complex, genetically and molecularly, I would say, engineered, precise um, organs or microprocessors. And the more we understand them, more precisely we can figure out where the problem is and how to fix it. So I think, at the deep level, we're beginning to understand brain not as a big organ with chunks of components, frontal cortex and big brainstem, etc., but much more as a complex sort of world that connects all the time and processes information. So that's understanding the brain at that level is really opening up our complete, you know, rethinking of neuroscience. I think the second thing that we're also beginning to understand is all of the mechanisms by which the brain is continuously reprogramming itself for plasticity, if we want to
Mm.
call that. Like every other AI system or generative AI or Chat GPT, it is constantly changing its information systems, its skill sets, and is learning all the time. So those two components are now giving us amazing tools to both understand diseases, but also develop new treatments for these diseases so that we can say, okay, how does um, molecularly targeted treatments affect these disorders? Or how do plasticity driven components of learning and rewiring help us treat diseases or understand diseases. So
Absolutely.
it's a fascinating sort of convergence of our what you might call digital understanding or this our understanding of the brain.
Yeah, absolutely. And uh, as you kind of walk through that, it reminds me of the conversation I was just having with Brandon Bensley over at Magnus about the SAINT protocol and how we're imaging the brain and better kind of understanding how to target it. Now, one thing that's been on my mind recently is uh, that seems to work for a let's call it a medical device setting. We don't yet seem to have drug um, capabilities that kind of work in such a precision way. Um, how are you starting to think about that? How are the kind of leading researchers starting to kind of approach that?
Yeah, no, that's a great question. And I think, we're just at a very early stage and to a large extent, very crude stage of understanding how to change these processes through therapies or other kinds of interventions. The classic example is plasticity enhancing drugs like ketamine. ketamine is a classic example of what you might say drug that affects not just a particular receptor or a particular mechanism, but actually creates a downstream change in the circuitry that would be long lasting, way longer than the presence of the drug in the brain. So ketamine gets in the brain and is out within a few hours, whereas its ET depressant effect doesn't show up for a day or so, and then lasts for several weeks, perhaps even. So that is the first, I would say, understanding and maybe a crude breakthrough to actually begin to figure out these mechanisms. Psychedelics have been another group. Again, these none of these are, what I would say going to be the long-term solutions for this mechanism because they all have various other effects and they're not necessarily clean drugs. Um, but this is opening up the field of understanding synaptic plasticity, understanding how brain rewires itself, learns itself, and how do we, through more precise molecular targets or drugs, could manipulate the system. So that's where I think would be the exciting part of treating chronic and severe mental illnesses and addictions.
You're starting to hint at this, and I'd like to maybe address it head on. Where do you sit on the side of the debate uh, around these psychedelics and whether the subjective effect is required, or whether you can just simply isolate the neuroplasticity effect?
Yeah, so far, I think, clearly people have not seen any compounds that do these types of, recovery without what I would say, the mind altering effects. So with ketamine, it could be dissociation. With psychedelics, obviously, it's essentially uh, psychedelic experience, if you will.
Mhm.
Um, so currently, the drugs that we are working with seem to all require that sort of, a trip, if you will, before it gets better. Uh, on the other hand, there are drugs that are beginning to be, I think, in the early stages of development that actually can look at the molecular mechanism of plasticity without activating some of the acute negative or mind altering types of symptoms. So, for example, one of the um, companies that one of the startups that I'm working with, we're looking at a compound that works directly through a modulation of the NMDA receptors in the brain, but it has a very strong ketamine-like antidepressant effect without any of the ketamine-like dissociative or acute effects. So there are beginning to be new compounds that are currently emerging. Um, let us see how well they turn out to be and if they actually are effective without causing a trip.
Yeah, absolutely. And I think that um, there are a lot of folks who are eagerly watching what we might call this second generation or next generation of
Right. It's the city.
neuroplastic. Yeah, exactly. I'd love to maybe just kind of hear a little bit more about your journey from research. I know that you spent some time in industry, starting your own company, developed one of the first novel MOAs for schizophrenia in quite a while, and then going back to kind of leading on the academic side. Take me to like the kind of break points in this journey and what kind of convinced you to maybe make those switches of what you were looking for.
Yeah, no, I mean, I think, I started out like most researchers and most academics, somewhat naively. I thought, if we do fundamental research and really discover novel mechanisms or discover, molecular pathways, then treatments will automatically come out. Well, it's and that's a lot of our initial kind of thinking. It's like, okay, if I study patients, if I study mouse models, I can come up with a way to understand diseases. I'll publish that, and then new drugs can be discovered from that. But what you realize is that that is a very long way from actually developing any kind of treatments, and that discovering, and publishing is really the first step in a very, very long journey. And unfortunately, that journey can take 20 years if we're, and even that is like 5% actually make that 20 year journey. Majority don't ever get to even start the journey after a paper is published. So that's really what frustrated me after about 15 years of really doing lots and lots of clinical trials and lots and lots of basic research. I just felt like I was in two separate worlds that never met. So that was when I started to think, how do we sort of connect the two? And I worked a lot with big pharma, and they do amazing things, but they also have a very sort of limited ability because of their business model to take lots of ideas into early human trials, because they have to really commit to a full scale development. So lots of good ideas I saw were being dropped by pharma because for a variety of reasons. So because that wasn't their focus, that was not a big market, that was a very complex disease. You can imagine many ways that we just haven't been able to do that. And that was the first experience for me with xanomeline for the schizophrenia treatment, where it was essentially going to be dropped by Lilly as an Alzheimer's drug. But we knew from basic research that this could potentially be a amazing schizophrenia drug. So that's really how I started to then sort of like borrow and steal from Eli Lilly to do that initial study to show the proof of principle, and of course now it's you know Cobenfy and all of that, a big story. So similarly, I kept thinking, instead of essentially waiting for big pharma to take these discoveries and try to bring up blockbuster drugs, I think it's very important for scientists to start thinking like, let us push it further in the commercial chain, whether it's a startup or whether it is a partnership. We should take the scientists know the science best, and we should push these commercial ideas further than we are currently doing. And that's really got me into the startup environment, started to think about many of the discoveries in our lab and others. How do we get it to, you know, even an IND stage, or even potentially it's, seed fund stage. So that's really sort of how I moved into more startup and entrepreneurial approach to it.
Yeah, I'd love to maybe go behind the scenes into the kind of decision to double down on exploring the applications in schizophrenia. Obviously, there wasn't a lot of um, let's call it positive action in this space. What gave you the confidence to pursue it?
I think there were a couple of things. One, as I said, there was tremendous amount of basic research that supported that this could actually be an effective drug. And a lot of it had to do with basic principles of how the muscarinic receptors can shut down dopamine transmission. So that was the real sort of strong scientific rationale that the dopamine transmission, which was predicted to be the expected thing in schizophrenia, and most of the effective drugs were dopamine antagonists. So there was very good sort of biological sense that if we can figure out a way to reduce dopamine without blocking the receptors, that it could be effective. The second thing that we also saw, and this was in the Lilly Alzheimer's studies, the compound, while it didn't really have massive effects on memory improvement, it did actually reduce the agitation and various what you might call um, antipsychotically responsive behavioral problems that Alzheimer's patients had. So it's sort of like, okay, if they can reduce their agitation, reduce their, even in some cases, this Alzheimer's patient had hallucinations, for example, where, sundowning and various other kinds of uh, mental uh, sort of symptoms, it improved those. So that gave us the confidence, certainly for me, that this was the way to do it. And the big problem was how to do it in a quick enough time before the drug was all flushed down the toilet by our belly. So we had 6 months, literally, to take the drug out of Lilly, and we had no money. We had to raise like a couple of 100,000. Fortunately, I had a clinical research unit where I was doing other clinical trials of antipsychotics in schizophrenic patients. So we were able to quickly do a 20-patient study within 6 months that and show that it was not only was it great for treating positive symptoms or the dopamine-driven symptoms, but it actually improved a lot of the negative symptoms and cognitive symptoms as well. So then I think you know, for a large sort of venture fund from Boston took the idea. Coronel Therapeutics was formed, and then, of course, eventually acquired by Bristol Myers.
Yeah. Well, you kind of skipped over a lot of the interesting part there. What was it like, getting to the kind of first proof of principle here and then, watching it just kinda take off from there?
Yeah. Well, I mean, I think the biggest, positive thing for me was to see that during that initial 6-month period, as patients were coming into the hospital,
Mm.
that we were able to discharge them within like 5 to 6 days.
Wow.
And within the first week, we would send them as outpatients and go home, come back as outpatients. And, most of these patients would be in group homes and various types of assisted living facilities. And these group homes would call us and say, wow, whatever you put this person on, please keep them on it because they are doing so well. So that was the kind of feedback we were getting. And then, clearly the big challenge was how do we then uh, get the compound reformulated, the compound made a little more safer through combination, and then doing a whole series of FDA enabling studies, which, as an individual investigator, I couldn't do that. You need people with business savvy, you need people, venture funds and various other types of talent that needs to be involved to take a compound all the way to market. So we were lucky. We found that with PureTech in Boston, and several amazing people got involved in this journey.
And, I've also had, Ray Sanchez from Cerevel on the show. I'm curious, what was the dynamic like watching the Cerevel, and Karuna kinda stories play out in parallel?
Yeah. Yeah, I mean, it was clearly, there was the very Cerebral had a similar kind of situation where they had shown a strong initial positive effect. And here was a muscarinic potentiator. And I think I'm still, you know, surprised that it didn't work as well as I thought it would in the large scale trials. And I think this is where there is I think, still a lot of learning that we need to have about doing clinical trials, especially large scale clinical trials in psychiatry. And we, because many of our measurements are subjective, rating scales and behavioral assessments, we have a lot of problems in terms of controlling all the variables in a study. If you're doing a diabetes study, it's very straightforward. You can look at blood sugar levels, you can look at hemoglobin A1C, and say, is the drug working or not? Whereas in schizophrenia or depression, it is so difficult to control and measure the outcomes properly. And what we did with Corona, and I would say this was like the team was amazing. Steve Brennan and a whole bunch of that did the trials. The biggest thing that we were able to do in those trials was, you know, very very rigorously control the assessment of patients, very rigorously control the type of patients we were enrolling, and the fidelity to the protocol across all of the sites. And if you don't do that, there's so much noise and so much variability that you could lose the therapeutic signal from the trial. Now, I don't know all the details of the average trial, but I just worry that in large scale trials, across the globe, that level of rigor could be lost. And even though the drug might be effective, because there's so many variables that are noisy within the trial, that you may lose the effect of the drug. So hopefully somebody will go back and read those studies.
It's funny. I ask a lot of people uh, what they would change or if they could change anything, what they would like to see. And this idea of, subjective scales and variability comes up a ton naturally because the placebo effect is so difficult to separate from in psychiatry. I'm curious, what do you think the kind of team at Karuna was doing specifically that was different in terms of the type of patients they were enrolling, protocol fidelity? I'd love to kind of go under the hood there.
Yeah. No, I think some of the big controls that we had was, first of all, we essentially replicated the same protocol that I did with 20 patients. We just did it 200 patients, 1,000 patients. It was essentially a simple, straightforward comparison study between placebo and drug, and nothing too fancy. So one of the problems that we try to do in clinical trials in psychiatry is that we try to ask too many questions from the drug. It's like the key question is the drug better than placebo? And you need to focus on your trial answering that question. But a lot of times what happens is people will say, well, what about the particular market segment we want to address? Why don't we enrich our patients for X? Or why don't we try to also discover a biomarker while we are at it, or a genetic signature? So you start to add all of these different nuances. What happens is that the study becomes very overburdened with questions and not able to answer the fundamental, purpose of the study. So that's the other thing that people should, not do. And then sometimes other structural things, such as, there's some evidence that having a placebo lead-in could be a problem in certain conditions like depression, or having sort of multiple assessments all the time, every 3 days. I mean, that's the other problem. People tend to measure things too many times. And then there's the practice effect on the patients. So if you ask the same question every third day, they start to then answer it differently than if you ask it once every 2 weeks, for example. So there's all this learning effect that then enhances placebo responses. So I think there's some foundational skill sets that people don't attend to. The other thing that we did was, all of the subjective measures, such as rating scales, have to also be recorded and then rescored by an objective person who doesn't have any knowledge of the patient. So that you need to sort of make it more objective than a doctor who is treating that patient,
Mm.
who, intrinsically wants to have the patient get better. So we tend to sort of look at their responses, over 4 weeks as improving as opposed to, you know, unless they're truly not improving or going in the wrong direction.
I love that. And I love the kind of push for simplicity. Um, I imagine there's like a temptation to find subgroups or um, a biomarker that's predictive. Um, when do you think that's appropriate versus not appropriate?
I think, those are appropriate after you've had, I would say, two solid phase two studies. And at that point, you have enough to know that you've got the fact that the drug works, or even a phase three trial. Once you've done that, then you can go back and refine it. It's like, who are the best responders? Who are the non-responders? What are some other biomarkers that might predict responders versus non-responders? But those are secondary questions and refining questions. And you shouldn't ask refining questions before you've got your primary answer. And that discipline is what's lacking, I think, in a lot of psychiatric drug.
Did you find you had to advocate for maintaining the kind of protocol fidelity from your original study? Like, was there temptation to follow like tell?
Splinty. One of the problems, and this is nothing against, great minds and top researchers, including myself and John, and we're all like that. But these companies, they have what I would say scientific advisory boards or key opinion leaders. And oftentimes those discussions end up doing exactly that because somebody feels like, oh, I have a genetic marker in schizophrenia. We should use that in the protocol. Or somebody else says, there's this great new study that came out that there's an EEG profile. We should use that. Or here's some study that said, don't use people with XYZ features, but use the other subtype. So these kinds of early discussions sometimes will distract the development of a simple protocol
Hmm.
because we all have our scientific biases and, you know, scientific advisory boards are notorious at making protocols complex because they are thinking of the science, not really this fundamental question of is the drug going to work or not? Or is it going to be better than placebo? And let's ask that simple question first.
Yeah, and I imagine everybody has a uh like a nuanced hypothesis they're also thinking about that they might want to tuck in. Yeah, fascinating. So what would you advise other biotechs to do vis-a-vis like engaging their advisory boards?
I would say, the advisory boards are great for understanding science if we're still in the early stages. If you have a proof of concept of a certain drug working, then make it simple and the advisory board can help you ask these refining questions after you've got the primary answer. And they're, of course, going to be important to sort of promote the concept or give scientific validation for the mechanism, or eventually even become key opinion leaders when you're going into market and etc. or FDA approval and various things like that. So it's an important component of drug development. But I think the use of that approach for the answering the simple first question can sort of mess it up. Uh, rather than help. And, I don't know about Cerevel, but if they can take that drug back and do the simple study that they initially showed positive effect on, I would love to see the results replicate.
Yeah, absolutely. Oh, I think we're all waiting to see what ends up transpiring. Uh, you mentioned this very quickly there. This idea that subjective scales make everything harder in psychiatry. Uh, why don't we have objective scales in psychiatry?
Yeah, I think we will sooner than later as technology is evolving. One of the problems, is that the brain, as you know, is a locked box. It's very hard to get to and study. And the imaging technology, EEG technologies we have had are relatively, I would say, simplistic or crude because they don't give you the level of detailed information we need to understand the microcircuits in the brain that may be involved in some of these, diseases or symptoms. I think that is beginning to change. People are beginning to now look at circuits and circuits for specific behaviors. Anhedonia is a good example where people are beginning to understand brain areas and even some neurotransmitters involved in um, and then some EEG signatures for various plasticity sort of outcomes. But as we refine those more and more, I think that's when we will have objective measures. Right now, they are sort of poorly validated and poorly refined. But I would say in the next 5 years, as brain imaging becomes more and more sort of sophisticated, and our EEGs, technologies, and surface recording technologies become more sophisticated, we will really get to sub-circuit level sort of signatures. And that's when we will have better biomarkers. Because genetic biomarkers, protein biomarkers are good for severe illnesses like Alzheimer's and other neurodegenerative things. But for psychiatric disorders, we need far more subtle and functional measures.
Yeah. Maybe, I'd love to hear, like, 5 years is not that far away. Where do you think these measures are gonna happen? Like, how do you think about, like, neurocircuitry as, like, a measurable, like, phenomenon or kind of, structure?
Yeah. No, I think the biggest thing that's going to open up that field is, I think, generative AI.
Oh, okay.
Because, the problem is the massive amount of data that we get, whether it's a PET scan or whether it's a 36 channel or 126 channel EEG recording. The amount of data and the amount of subsets within that are so complicated that you need massive data processing to understand. It's kind of like the whole genome sequencing. Without AI, we can't make head or tail of whole genome sequence data.
Mm.
In the same way, brain imaging or brain recordings are so complex and so many vectors that you have to really disambiguate. You need massive computational as well as generative AI programs to do that. But I think that will happen if we can, you know, uh, use this emerging massive amounts of data processing, speed, and complexity of calculations. I fully see in the next 5 years AI will crack the code.
So it's in your, like, estimate, it's a computational and data processing problem, not a measurement problem.
I think there's some measurement problem, but the measurement problem is really because of the sensitivity of analysis, so that you know you can measure things, but when like for example, uh, I would say cosmology is a good example. The signals are so faint coming from let's say 10 billion light years away. Um, but now we can actually visualize a black hole because we can not only pick up those tiny, tiny signals, but then also recreate what they're actually telling us. And I see that's what is going to happen in brain, you know, AI driven brain signal analysis.
Fascinating. So while we're on the topic of the future here, I'm curious to hear, like, what else are you most excited to see? What other areas of research are you watching most closely?
Well, I mean, I think the other area of research that is truly exciting is all of the ways we're beginning to sort of recreate cellular mechanisms um, in synthetic ways. So synthetic biology in the sense of creating organelles, creating, you know, gene therapy is the crudest way of that. But we imagine we could actually begin to create new lysosomes or new mitochondria and begin to sort of address all of these very complex cellular mechanisms in a synthetic way. That would be the ultimate for medical interventions.
I'm too curious not to go there. I haven't heard anybody talk about synthetic biology with respect to psychiatry and neurology before. Where are the applications that you're excited to see?
I know, yeah. So, and a good example, again, this is science fiction now.
We'll do a science fiction segment of podcast.
Yeah. So, but imagine bipolar disorder is a mitochondrial disorder. And the thing that might be happening in such a disorder is that there is fluctuations in mitochondrial regulation and energy metabolism. So when people are manic, they have so much energy, so much ideas. They don't need sleep. They don't need food because the mitochondria might be ramped up to a point of not needing anything and producing so much energy. Whereas on the depressive side, they would be the exact opposite, where the mitochondria now create very little energy, and you are just kind of sleeping all the time, but can't think straight. Imagine if that were the case, and we could sympathetically change that and make it even peeled. Um, that could completely change the way we approach bipolar or some other genetic metabolic disease, for example.
Yeah, instead of trying to go to the uh, I guess like the root gene kind of source, going to maybe like a more complicated, complex kind of structural um mechanism that might be more closely tied with the outcome. Yeah. Yeah. Um yeah, gene editing has so many leaps and bounds uh before it actually has the effect in a lot of cases.
Exactly. So
Fascinating.
okay, that would be, I don't know. That's a probably a 30-year solution. I'm not sure that that's going to happen in the next 5 years.
Yeah. Do you see that the practice of psychiatry and neurology needing to evolve given the kind of development of some of these new interventions and like interventional psychiatry in general? Like how do you think the practice is going to have to evolve?
Yeah, I think one of the things that unfortunately we have not you know done is continuous education as neuroscience is evolving so fast that the clinical practice of psychiatry still is in the 90s, and we are still focused on prescribing medicines as the primary sort of and the medicines are all basically 50 years old in terms of the principles. They've not been anything new has come out that's taken advantage of some of the advances of the neuroscience yet. So I think in the next 2 decades we will have to see that there's a much more rapid transmission of knowledge to clinical practice than it is current. Because right now, people are still prescribing drugs that they learned as residents 40 years ago.
How is this changing your your kind of educational philosophy? At the yeah, as the dean, you have control now, no?
Yeah. Well, sort of, but yeah. But no, absolutely. And I think that is really the big challenge that we all face is like we need to make psychiatrists and neurologists much more, I would say, practicing neuroscientists rather than research neuroscientists do their thing and then practicing clinicians, you know, do whatever the drug company tells them to do. That's the kind of dichotomy we need to completely eliminate. One of the big problems, I think, also is that we need to change our accreditation agencies, expectation of licensure, continuing medical education, especially in a rapidly evolving field like neuroscience and psychiatry. Uh, you know, appendectomy has been the same for the last 200 years. And yes, we've gotten to do it now with minimally invasive techniques and all of that. But the basic principle is still the same. But that's not the case with neuroscience and psychiatry. So we need to keep moving faster, quicker into clinical implementation.
Yeah, fascinating. Do you do you anticipate neurologists and psychiatrists in practice converging, or is it more of like a baseline, understanding converging?
I think they will completely converge. I really, I mean, that would be my certainly my hope, but I think my prediction. Now, what will happen, for example, even within neurology as a just a general field, there are interventional neurologists who are essentially neurosurgeons, right? I mean, they go in and put the, probe into the carotid and pull out the clot. They're quote neurologists, but for all practical purposes, they're neurosurgeons. But they don't have to go through a neurosurgery residency. So, do we say that, a person who treats Parkinson's disease in a typical clinical setting versus this person who's in the OR all the time pulling down clots from carotids are the same subspecialty? Officially, they are, but, you know, functionally, they're nowhere near each other. I see the same thing will happen with psychiatrists and neurology. People who are going to treat acute strokes will be very different than people who are treating acute psychotic episodes versus people who are treating, chronic OCD, for example. So I see this emerging and there will be a continuum of people who are dealing all the way, much like rehab medicine to stroke to neurosurgery right now. So I see that. And some of these board expectations and our residency expectations will start to bleed into each other. And eventually, 50 years from now, I hope there will be 5 subspecialties and not 50.
Yeah. Oh, so fascinating. I feel like we could talk about the kind of evolution of clinical practice for another hour here, but we sadly don't have it. Um, uh, I'd like to maybe kind of close by bookending our conversation. You started your career due to a kind of personal experience in the family with schizophrenia. You're actually a core part of seeing the first new development in the space. Did you anticipate that it would be that your career would be so directly linked to the starting point? Like how did that feel?
It feels amazing. It's like a dream come true. How can one possibly imagine? This is why I went to medicine. Yeah, literally 40 years ago. And the first ever new treatment for that disease in 75 years, literally, is coming out of the work I did. I mean, it's just a great, amazing, humbling feeling.
Yeah. Well, it's a feeling I'm sure a lot of people who are listening to this show are chasing. Not a lot of people get to experience, and congratulations.
Well, thank you. Thank you. And I wish them all the best. I hope everyone succeeds in that kind of dream.
Absolutely. We're all on the same team in that sense.
Yes.
Yeah. Well, thank you so much for taking the time. I really appreciate you joining me on the show today.
Yeah, it was delightful talking to you, Brandon, and I love your show. Thank you.
Yeah, of course.